Hi, welcome back. So now we're going to go over protein synthesis. I already did a video reviewing RNA and DNA. So if you're not remembering much about RNA and DNA, I highly suggest you go view that video and get a refresher on it, okay? But this video is all about protein synthesis. So what is protein synthesis? Protein synthesis is how the body or how the cells make proteins. This is very important because the proteins are the doers of the cell. If you don't have proteins to do the stuff in the cell, then nothing's going to get done. What happens if nothing's done in the cell? Nothing's done in the tissue. If there's nothing done in the tissue, nothing is done in the organ. If nothing's done in the organ, nothing's done in the organ system. If nothing's done in that organ system, it's not done in the body. If it's not done in the body, what happens to the organism? It doesn't do anything either. So you have to be able to make proteins. Protein synthesis is the assembly of amino acids to form proteins. Now wait a minute. I said it's how the body makes proteins, right? Not the assembly of amino acids. Well, proteins, if you remember from when we did biochemistry, proteins are made out of amino acids. So you have a bunch of amino acids. I'm going to pick leucine, because it's honestly one of my favorites right now, right beside met. I'll include that one since I talked about it in ET, which is not where met goes, but that's fine. Well, when you assemble in a line of amino acids, you form a protein. This is what proteins are made out of. They are bound by these bonds called polypeptide bonds. This is really important because proteins are also called polypeptide chains. We'll come back to that in just a minute. Now there are two steps to protein synthesis: translation and transcription. Those sound very, very much alike, right? Well, this is where remembering those analogies I made to RNA in the first video are going to be very helpful. Transcription is all about mRNA. That's the only type of RNA that we really talk about in transcription is mRNA. And what transcription is it's the copying of DNA to mRNA. So if you'll remember from history, I'm sure you've heard sometime of something called a scribe. Well, what scribes did is they kept a record of things going on. They knew how to read and write, and they would write down all the important things that happened. Well, in transcription, we are acting as a scribe in writing down the important information in your DNA into mRNA so it can travel outside the nucleus. To relate this to my cookbook analogy, inside of that cookbook, I've opened up my cookbook, I've found the recipe or the gene that I want to transcribe or write down. Now I'm going to take that recipe, my gene, out of my DNA, and I'm going to write it on an index card. That index card that I'm writing it onto is the assembly of this mRNA. And then I can close the book and put it away, take my mRNA or my index card with the recipe, and go anywhere I want with it. I can send it to my mom. So that's what transcription is. Then translation. Translation is when we have all three different types of RNA. Now, this is a little wordy, so I'm going to break it down. Translation is the RNA reading the mRNA code and assembling the amino acids in order to form a polypeptide chain protein. All right, to break that down. RNA, I'm going to put parentheses around it because ribosomes are made out of RNA, rRNA. In the nucleolus, rRNA is produced to form ribosomes for protein synthesis. We talked about that when we talked about the different types of cell organelles. So the rRNA that's produced forms the ribosomes, which we need because the ribosomes read what is written on the mRNA. If any of you have really seen my handwriting, and all of this kind of shows you how bad that really is, when my mom gets my recipe, she is going to have to read it, and she's going to have to translate it so that she understands what it's saying, because my handwriting can get kind of messy. So my mom is going to read the message or the recipe that I've written, the messenger RNA, and she is going to assemble the amino acids that she needs. Remember that the tRNA is shaped like a T? Up here it has the amino acid. I'm going to abbreviate that to AA to make it easier on me. Well, the amino acids are held up here. Down here, we have our anticodon. I think AUC is the one I've been using. So, here's our tRNA that we need. You see how we have all three types of RNA we're using here? Yep. And here's that polypeptide chain. It's coming back up. Remember that our amino acids, and I'm abbreviating again, are held together by this bond. That is a polypeptide bond, and when you get a lot of them, it forms into a chain. So a polypeptide chain is just another word or another way to say protein. They're synonym. Okay. Now let's see if we can go through this and make all of that red a little bit more clear. Transcription is happening inside the nucleus. So we're inside the nucleus. The mRNA is going to read the DNA strand. So remember inside the nucleus we have DNA, right? Double-stranded and held together to protect it. Well, what it's going to do is we're going to have a strand of DNA. And I'm just going to use 4 here, A, T, C, G, because this is DNA. The DNA is going to have a complementary strand of DNA. That would be T, A, G, C. They're held together by hydrogen bonds. RNA polymerase is going to come in and break those hydrogen bonds and pull the complementary strand of DNA away. When it does this, the mRNA has a chance to come in and create a complementary strand of mRNA. So while the DNA polymerase is holding it apart, there is going to be a piece of RNA that comes in that is a complement to that A. For RNA, it will be uracil. Then another piece will find it's a complement of T. That's going to be A. Well, these two are going to bond together to form RNA backbone. The same thing will happen with C and G. Just like that. Now we have a piece of mRNA. This RNA is going to leave the nucleus to find a ribosome, again, made out of 2 parts. And this is where translation is going to happen. So let's take a quick look at how transcription works. First, the DNA strand is going to split to expose the active strand. This is the active strand down here, and it has split. These hydrogen bonds have been broken. The complementary mRNA nucleotides are lining up opposite of the active DNA strand. This T was complemented by this A. That's where it started. Then this A with this U, this C with this G, and so on. This is where the RNA polymerase is currently at. And this is where we are matching things up and moving them out. The mRNA will leave the nucleus to find a ribosome. Something I want you to look at right here real quick is that these are the two that were just matched. So now another piece of RNA is going to be in the nucleus. It wants to match up with A. The complement, U. It's going to float in here, match up with this A, and join on to the mRNA. Then when it's finished copying this gene, it's going to move out of the nucleus to find a ribosome. Once it finds a ribosome, that's when translation occurs. Translation happens outside of the nucleus. It's in the cytoplasm on a ribosome. And all three types of RNA have a job to produce here. So I'm going to sketch out a ribosome really quick. Again, very vague drawing. So in this ribosome, remember that it's made out of rRNA. Well, it's going to receive a strand of mRNA in here. We're going to use A, U, and C. And of course, it has more bases down this line. Well, when it gets to this active spot here of the larger portion of the ribosome, a piece of tRNA is going to come in. Remember that it's going to have an amino acid up here. These three bases of mRNA are called a codon. And believe it or not, but they match up with an anticodon. This is supposed to be a C. The anticodon is going to be complementary. So we're going to have a U, A, G for our anticodon. That means that this is the specific amino acid that needs to come right here. Once we have that, this amino acid is going to be held on tight by the RNA. This piece of tRNA is going to leave, and a new one is going to come in to match the next three bases or the next codon. The same process will happen again, and eventually you're going to have a long amino acid chain. It will eventually form its final three-dimensional structure after it's been released from the ribosome. So, how it works. Ribosomes decode the mRNA. They do this by using sets of three bases called codons. So every three bases on an mRNA is a codon. Each codon is matched to its anticodon on the tRNA. This shows you what a tRNA looks like. Down here in the red, that's the red is the anticodon, the green is the codon. And up here is where your amino acid is held. The tRNA holds an amino acid which is bonded to the other amino acids by the ribosome. So here you can see you have three spots inside of the ribosome where the tRNA comes in. At the beginning, there are pieces of tRNA just floating around. They come in and attach here. They will go to this next spot where their amino acid is chained onto the polypeptide chain. And then they leave. And that's translation.